Hello, everyone. Thank you. So today I'm going to chair this exciting session on challenges in drug discovery. And to set up the ground, I prepared a few slides. This morning we already touched on one of the biggest challenges in our field, which is the EROM law, something we need to maybe put on the graphic. And I want to start with the classic Moore's law. So Moore's law, basically, this is a progression of computer hardware. So the trend holds from early 70s, indicating that computers become cheaper and more powerful every couple of years, and they grow up exponentially. And this exponential growth of computational hardware drives the progress on the planet in many other fields, especially heavy on high tech. So everything is exponential right now. Everything successful is exponential right now. With one exception, this is our area, drug discovery, which is exponential in the reverse. That's why this is called EROM law, which is more spelled in reverse. So this is the number of drugs one can develop for $1 billion. As you can see, the bar is below 1, because now it's around $2.6 billion to develop one drug on average. And this trend, again, probably it's slowing a bit, hopefully, but the trend is really staggering. And like Jerry Claude mentioned this morning, I love this quote that if something cannot go on, it won't. So the big question is, what is going to happen with the EROM law? How our industry is going to progress, or is it? And I'll just do some shameless promotion. Just yesterday, there was a drug discovery today review by myself and Alex Dropsha came up where we actually discussed these things. How are we going to reverse the EROM law? If it's even possible, what's going to be done? How it's going to happen? I'm not going to go into details again. Self-promotion, you go and read, please. Idea being that we have tried to apply the concept of 6Ds. This is a concept which transforms the modern world in digital disruption way. Again, without going specifics, in order to technology become exponential, it has to go through 6D stages: digitization, deception, disruption, demonetization, dematerialization, and democratization. And by what we kind of came up with, drug discovery right now going in the last D through democratization. That's what is happening, and that's what is needed to reverse that EROM, to make it more, to really reverse the trend. And the today's session is pretty much, and this conference is about this last D, democratization. Because one of the key messages and drivers of democratization is open science, open collaboration, competitions like cash. So if this is right, then in fact we are participating right now in making a history. We are pushing our science through democratization critical final stage. And cash was a great example of that. And our first speaker is Matthieu Schapira, who is one of the key organizers, the organizer of cash competitions. And he is a professor of toxicology and pharmacology at the University of Toronto. He is a chief computational biologist and bioinformatician at Structural Genomics Consortium. 1 year ago, give and take, Professor Schapira gave us results of CASH 1 when we met in Toronto. So today we're going to hear about the results of CASH 2 and 3. We're going to see where the field is going, and we're going to discuss how we all can contribute to this democratization of drug discovery, which again, hopefully, due to our collective efforts, will make the situation different. So Professor Schapira, the floor is yours. Thank you.
Thanks, Art. Everyone. All right, so I'll tell you about cash. I believe many or most of you know that the cash was modeled after CASP, the protein structure prediction challenge that set the stage for a breakthrough in the field in protein structure prediction. And similarly, the goal of cash is to set the stage for a breakthrough in computational hit finding. We are scrupulously following a roadmap that was delineated back in 2022 by this group of folks, both from academia and from the industry. And I guess the defining factor of cash probably is that it is a prospective hit finding challenge. So every 6 months or so, a new challenge is launched. where participants are asked to use their method to predict up to 100 molecules that are then ordered and tested experimentally at the Structural Genomics Consortium in Toronto in the lab of Cheryl Arrowsmith with a biophysics platform that has been used for the past 15 years to develop chemical probes for dozens of targets with numerous pharma partners. So it's a platform with really state-of-the-art equipment and people who are very experienced in testing small molecules. At the end of each challenge, all data is publicly released with no restriction. We encourage as much as possible participants to make their method open source, but we incentivize them to do so as well. But we do understand that, for instance, biotech companies are not necessarily open to this, but they definitely are asked to provide a description of their method that is sufficiently detailed that an expert in the field can learn from this. And at the end of so during the challenge, the participants are anonymous, and the top performing groups at the very end are de-anonymized. So today, when a scientist reads a paper on computational hit finding, or a pharma company hears a sales pitch on computational hit finding, it's not easy to compare these different methods that are all focused on a different target and confirmed predictions are confirmed experimentally with different assays on different hands. And so here the idea is for CASP to provide a unifying metric where all of these methods are tested on the same target and confirmed experimentally with the same assay and the same hands. The first CASP challenge was focused on LRRK2, which is the most commonly mutated gene in familial Parkinson's disease. Pointer on this. At any rate, and so there were known compound binding the kinase domain, quite advanced compound binding the kinase domain of LRRK2, but none of them binding the WDR domain. And so the challenge here was to find a false molecule that would bind the WDR domain of LRRK2. And CASP participants did deliver the first such molecules. I'm just showing some of them here. Here is a general overview, if you will, of the different computational methods that were deployed by the participants in this first challenge. For instance, the first one at the top here, participant number 1187, so workflow WF1187. They just used one method, which was a deep learning docking, and the output of this was their selection. One step. Well, the second participant here studied with an ultra high throughput docking, physics-based, and the output of this was further refined using a deep learning docking method. Of course, I'm not going to go over all of this, but suffice it to say that there were like 3, 4, 5, 6, 7 steps. So you can see that participants combine physics-based with machine learning methods in multiple different ways, exploring different avenues, and I think this speaks to the fact that that right now the field is very much in an exploratory mode, where we are still not clear which avenue is the most efficient for computational hit finding. And therefore, the merit of benchmarking exercises like CASP and others, where we really are trying to compare these different methods, provide a unifying metric, and try to define the state of the art to try to guide future progress. Each CASP challenge is composed of 2 rounds. A first round, round 1, is what we call the hit identification round at the very end over there. And so, for instance, here you see that this team came up with this was one of their 100 or so molecules that were predicted, And you can see that the binding affinity, the binding affinity of this molecule is weak, 120 micromolar or so. But it doesn't bind to the unrelated target, NSD2, which was a good sign. However, there was a big red flag for this molecule, which is that it shows signs of insolubility and aggregation at 200 micromolar, which all in all is pretty close to the observed binding affinity. And if you look at this, probably someone if this were part of the real drug discovery program, probably medicinal chemists would say, I won't touch this with a 10-foot pole. I have much better compounds elsewhere, and I don't want to waste resources on this. And this is a luxury that we do not have in cash, because we cannot take the risk of saying, well, I don't believe in this molecule, and probably this method didn't work, and therefore we should dismiss this computational workflow. So instead, what we do, of course, we avoid as much as possible false positives, so we try to characterize this with multiple assays, but we also try to avoid false negatives. And the way we do this is we go back to the participants, say, okay, this is your hit. We don't tell them, but we don't believe in this hit. Nevertheless, you can go ahead and order up to 50 chemical analogs of this molecule. And this is what happened here. So these participants selected 33 analogs. And out of these 33, 7 turned out to be active. Included this, I'm just showing one of them here, this molecule, which in this surface plasmon resonant assay binds with a KD around 50 micromolar, so a reasonable improvement. But importantly, you can see that this compound passes solubility and aggregation measurement by the dynamic light scattering. So it's behaving well in solution at 200 micromolar. And the cherry on the cake here is that this compound also had a fluorine. And therefore, we could use a F19 NMR assay as a secondary orthogonal binding assay that again confirmed that this molecule was binding to LAR2. So now this is signifying that probably the binding signal is not assay specific, but this is a real hit. So we went from a molecule that were very sketchy to a chemical series that is pretty well established. But we don't make this call. We don't say, oh, no, we believe strongly in this. There is an independent hit evaluation committee that makes the call, and it is composed of 2 biophysicists, Anders Gunnarsson at AstraZeneca and Vera Putter at Nuvisan, medicinal chemist Hartmut Beck at Bayer and Lars Wortmann at Boehringer Ingelheim, and as well as a computational chemist, Pat Walters at Relay Therapeutics. So these are people who are keen-eye to identify assay readouts or binding signals that are dubious, people who have a forgiving eye to rule out compound with chemical liabilities or shaky SAR, etc. So the data, the experimental data is not easy to interpret, especially when you are not an expert in the field. And therefore, we're also providing, when we provide the data, we also provided some kind of manual to try to explain what the data means, what these sensograms mean, what this saturation curve mean, how you calculate the KD value, and also importantly, not only the actually readout data, but the metadata that provides some indication on the quality and the reliability of the binding signal. And for instance, if you could receive the data on your compound and with a KD associated with your molecule of 20 micromolar, say, yeah, hooray, I've got a great hit. But you also need to look at the chi-square, and if you see that the chi-square is greater than 10% of your Rmax, probably it's a sign that this KD value is not reliable. So we do this to be as transparent as possible, understanding that it can add confusion, which is why we provide a manual and also provide, in addition to the long Excel spreadsheet with all the data, some PowerPoint with the take-home message. So what were the most successful strategies in the first cache challenge? So 6 out of these 7 most successful strategies had some component of machine learning in them. 5 of them, some step using deep learning at one point or another. 2 of them were plain virtual, not plain, but where systematic virtual screening, where each molecule is tested one after the other from a commercial library. In this case, it was there was some active learning from actually this group was the Art Cherkasov and Francesco Gentile. Well, this group here used deep learning to score compounds. And a number of groups, including Dmitry Kiryev, who is also somewhere in the room, used a more de novo generative design approaches, either to link or grow fragments, or to grow smiles using reinvent a well-known machine learning model. And finally, there was one purely physics-based method that did very well. It was kind of a brute force where the virtual flow was deployed on thousands of CPUs available at Harvard to Christoph Gargula. At the end of each challenge, we write an overview manuscript where we invite all participants to join as co-authors. Typically, those who do well said yes, happy to join, and those who did not, no, thank you. And participants also write their own papers, and here are some examples for the first CASH challenge. And I think I'm going to move on now to the second CASH challenge. I'm going to give you a few vignettes on the different challenges, and then I'll conclude with some general observations. So CASH 2 was focused on helicase, SARS-CoV-2 helicase, called NSP13. So the way this enzyme works is that it hydrolyzes ATP to produce the energy necessary to unfold RNA duplexes. So one of the RNA strands is sieved through the core of the protein through this channel, while the other RNA strand is wrapping around, is not shown here. And at the time this challenge was launched, there were 4 compound fragments that had been crystallized bound to this central RNA binding channel. These fragments have no measurable binding affinity, so extremely weak, captured crystallographically. And so the challenge was to find molecule that bind here with with measurable binding affinity. And by the way, this is the most conserved binding pocket in the coronavirus proteome. So, of course, there is some relevance there. So, 23 computational teams completed the challenge. Collectively, they selected close to 2,000 compounds that were tested experimentally. And this produced a dozen hits with double-digit micromolar binding affinity, and I'm going to show just a few of them here. Excuse me. So the compound that had the best score from the from the hit evaluation committee was this molecule here. So you see that it's binding with an affinity of around 30 micromolar, which is not bad for it's a pretty small molecule. It must be binding quite efficiently. And it was specific versus an anti-target. It was binding also not only by SPR, but F19 NMR. And in the dynamic light scattering experiment, it was soluble, no sign of aggregation, so all good. What's fascinating about this molecule is that it was actually designed by a gamer using an online interface called Foldit. The way it works is that these citizen scientists, if you will, are provided with the structure here, and online they can use this structure. Online they can use this structure to add atoms or functional groups here and there on the fragments. After which, in the background there is a tool that looks for commercial analogs, positions the commercial analogs in the pocket, and then they can decide to reject or accept the pose. And after multiple such cycles, they come up with this compound. And the organizers added at the very end some deep learning scoring function to finalize the selection. So here it looks like combining the creativity, if you will, of these gamers with in the background this well-known computational tools was some kind of winning combination. This compound here was one of several from Karina Machado's group team. Karina is somewhere around here, and we'll be presenting this in detail tomorrow. Again, binding well, confirmed by F19 NMR, behaving well in solution, and so the way, if I want to summarize very briefly this method, so they first generated this very diverse library of, I believe, 10,000 compounds only, docked it with a regular open source physics-based virtual screening protocol, and used this as a training set for a machine learning model that was used then to screen very rapidly millions or billions of molecules. And then there were some further refinement steps that Karina, I'm sure, will tell you about tomorrow. And the last vignette here, these two molecules, obviously chemical analog from this team, the Gennady Poda's team at the Ontario Institute for Cancer Research, who used purely physics-based methods. So they used GLIDE, which has been the gold standard in virtual screening for the past 30 years. And then they refined this with HIDE, which is a tool from Biosolvit to account for water molecules. And evidently, when used by experts and seasoned computational chemists, these physics-based methods seem to produce good results. I believe the 10 most potent compounds from cache 2 were also sent to Carla Satchel's group at Northwestern University, who had an RNA unwinding assay. And so they tested these compounds. We knew they were binding, but we did not know whether they were inhibiting the catalytic activity. And two of them did inhibit the catalytic activity. This compound from the Karina's group, and this compound. Well, this compound was by far the most potent, by a KD around 700 nanomolar, and in the C50 500 nanomolar. However, this compound was not scored well at all by the heat evaluation committee, and many of you, of course, will be horrified to see this ester group in the middle, among other things. So probably this compound would break into pretty rapidly in cells and in other systems. So that was one of the reasons why this compound was scored poorly. Probably another reason was that this compound was a singleton. Indeed, so if you remember, we have two rounds, and so the parent in quotation mark, the parent molecule for this hit is on the left here. So this is the compound, one of the compounds selected by the Kunwan Park team. And when asked to select analogs to confirm this compound, they ended up with something like this. So I guess you need a lot of imagination, creativity, I guess, to go from this to calling this an analog. But somehow they did amazingly well, it seems, going from this to this 700 nanomolar compound, and it's binding by F19 NMR, it's binding by SPR, it's inhibiting the enzymatic activity. So we don't really know what to make of this compound. Is it real? Is it not? If it's real, it's actually a quite interesting molecule. And there might be some medicinal chemists who walk around starting with replacing this ester with an MI group, for instance. So it makes you wonder, is Kunwan Park a genius? Maybe he is. And another question that is coming from this is, how strict should we be on the selection of analogs? If someone is sending us this as an analog of that, should we tell them, oh, sorry, we can't really make any conclusion out of this? All right, I'm going to move on to cache 3 very quickly. So cache 3 was focused on NSP3, another coronavirus protein that helps the virus evade the immune system. And so here's the macro domain of this protein. and the challenge was to find a molecule that would compete with ADP ribose. Much after this challenge was launched, actually about six or 8 months ago, I want to say, Jamie Fraser's group at UCSF published this preprint showing that where they developed a quite potent molecule targeting this very binding pocket, this very site, and they showed that their molecule could reduce viral replication, potentiate innate immune response, and lead to a survival benefit in animal models of coronavirus. So it seems to be really that there is something to this target, which is actually very, very rewarding, and it's really great to see that we got many computational groups and AI experts focusing on this target that seems to be actually a pretty interesting target for SARS-CoV-2. But at the time this challenge was launched, this was the landscape, if you will, of the chemical coverage of this protein. So this, again, generated by Jamie Fraser's group, who was our target expert for Cache3. So whenever we had scientific questions on the target, he was here to help us. And so at the time the challenge was launched, there were two types of molecules, I want to say. Quite potent molecules at the bottom here, but they all had this carboxylic acid that was identified as a chemical liability. This compound didn't translate very well in cell assays. Or this type of molecules that did not have a carboxylic acid, at least some of them, but were less potent. So the goal here that we set out for participants of Cache3 was to find novel scaffolds that would not have, that would return sufficient potency, but without the carboxylic acid. And I'll just give 2 or 3 examples here. So the first one here was discovered by Francesco Gentili and Cherkasov, where they used their deep dock method. They were pioneered this approach, deep docking, where you use docking scores from physics-based virtual screening as a training set for a machine learning model that can then screen billions of molecules, and then they refine the results with consensus docking and consensus scoring. And Francesco will tell you more about this tomorrow, so don't miss Francesco's talk. And so this was the winning molecule according to the hit evaluation committee. In their view, this was a sufficiently novel molecule because this was what was in the PNAS paper here. And you can see that it was not obvious, not trivial to identify this amide group here, mostly, and a little bit of change also at this level, to go from this pretty weak fragment to a much stronger fragment. And the compound was also crystallized by Gal and Corey in the Fraser group. Minghu Song at U Connecticut ended up using a different path to the very similar type of molecules. Again, amide bond on this bicyclic system with some changes also on this ring. And here what they used was a purely physics-based method, high-throughput docking, looking for a diverse set out of the output, molecular mechanics, free energy calculation, and electron density calculation that I'm less familiar with, but it worked very well for him. And again, crystal structure from Galen at UC in Jamie Fraser's group. Edina Rosta at UCL found many hits, but they were judged less innovative and less novel, I should rather say, but they were pretty potent and crystallized as well. And the last example is this molecule, which by far was the most novel, much more novel than anything else we got. With a KD of 30 micromolar, the molecule seems to be a little bit sticky. It doesn't come off the protein or the chip fast enough. But it was selective against the anti-target. It was behaving well in solution. It was active in the HCRF displacement assay. So a lot of good encouraging results from this molecule. But again, it was not scored highly by the heat evaluation committee, probably for two reasons. Again, maybe there are some chemical red or orange flags. For instance, some chemists don't like furan group or other things. And the other reason was, again, this was a singleton. And so this was the parent molecule. So this was this molecule. And when we asked for analogs, these are the molecules that were sent to us as analogs, which they are not. And so, again, this molecule, again, comes from the Kuhn-1 group. So, again, in cache 2, they came up with the most potent molecule. In cache 3, they came up with the most novel molecule. And in both cases, it's a singleton with some potential chemical liability, and we don't really know what to make of this, which makes you, again, wonder, is Kuhn-1 a genius? Makes you wonder. Anyway, in terms of computational methods, so they started with a random forest classification, probably using the compound that at the time were in the PNS paper, and then it's a pretty standard physics-based pipeline, high-throughput docking, MD, free energy calculation, diverse set, visual inspection. So, so far we have 6 cache challenges that 3 of them are completed. You can find all the data on the cache website. So we have a paper on this one, a preprint on this one, preprint in progress, I mean manuscript in progress on this one. So this one will release the data in, I believe, a month and a half or so. And if you want to know more, you can look at this editorial that was published last year. So some of the lessons. First, a big lesson for me was expect the unexpected. As I have mentioned, so the best scoring compound was discovered via crowdsourcing, where gamers used an online interface within the background and Rosetta ligand, and the organizers that use neural network scoring function to finalize the selection. The second example is the best hit in cache 2, the best hit rate, sorry, not the best hit rate in cache 2 was from Carrera Machado and her colleagues. At the time, I don't think they were really on the map on computational chemistry. I think now they are much more so. So it's great to see that we are able to shine the light on people who make important work in the field, important contribution. In terms of operations, in terms of running these benchmarking challenges, something that was very helpful to us was to have this very clear roadmap at the outset that was delineated by this group of experts. For instance, for me and for many of us, actually, the heat expansion round at first, the merit of this heat expansion round we were not sold on this. And it turned out it was very important to build sufficient confidence on the hits that we are observing from the first round. The involvement of industry in the governing board and in independent scientific community adds rigor and I believe credibility. Rigor in all sense of the term, rigor in terms of, well, we're doing things really properly, but also sometimes it feels a bit constraining. You know, sometimes in academia we feel like, oh, well, we're getting overexcited about this or that. So rigor is here and it's here to stay, and I think it's a good thing. Something that definitely we were not good enough at the beginning. I think the team at Conscience is helping tremendously with this, is communication and transparency with participants. And I think thanks to Conscience, we'll do even better than we have so far. And in terms of science, some of the lessons, yes, the field is highly exploratory. We see all this combination of methods that are going in all possible directions. And so far, we are starting to see some trends across different challenges. some of the strategies that seem to be recurrently successful, at least in some people's hands. The first one is to use slow physics-based methods to generate training set to train fast machine learning models in active learning cycles. The second, so it's a mechanism, of course, to screen very efficiently very large data set. Another trend that seems to be working well is generative design, especially fragment-based so far. And so this generative design, of course, is a much more elegant way, if you will, to traverse the immensity of the chemistry space. And then, but then you have the constraint of, once you have inventing a novel compound, you have to the constraint of trying to identify a commercially available analog, because we do not include custom chemistry in cache. And definitely purely physics-based method remain competitive at this point. This said, we also see that the hits that are confirmed experimentally are all in all quite rare and often, not always, but often weak, which speaks to the fact that an AI-driven breakthrough remains to be seen. So I'd like to acknowledge the hard work of my colleagues. A big shout out at the biophysics platform at the SGC, especially Sasha Herasimenko and Madushika Silva, who are leading the charge. Dakota 311 at Conscience is doing an absolutely amazing job making sure that things are running smoothly, and we would be in a bad shape without her help. And the whole Conscience team, of course. And of course, the contribution of the governing board and the different committees is essential. And finally, yes, so big thanks to Conscience, who is playing a leading role in operations, SGC in testing the compound and the scientific aspect, and these different funding organizations. Thank you.
Thank you so much, Matthieu. All right, so now we're going to hear about another exciting open science initiative, and the presenter is Jenke Scheen, who is a senior scientist at ASAP Discovery and at the Open Molecular Software Foundation. And we are going to hear about one of the ASAP Discovery initiatives on antiviral development. So the floor is yours. Here's the mic.
All right, thank you. Okay, so hi everyone. First of all, I'd like to thank the organizers, especially Dylan, for inviting me to speak here today. I'd also like to compliment them on the quality so far of this conference, and also I think it's really a mark of a well-organized conference if the talks before your own talk kind of talk about a lot of things that make me not have to do a lot of my introduction. So that's great. And also if slides are repeated. So my first slide has been mentioned twice already, I think. Which is great, so I don't have to explain any of this anymore. The point that this particular slide is trying to make, this is just a variation on the plot that we've seen before, is that there is the hypothesis that computational chemistry could potentially save us from Irem's law, right? So the two things that we'd want to do to kind of solve this problem is either reducing the cost of making a drug or making more drugs, right? That's just how to resolve that ratio. And as I mentioned before, in many talks of so far, it's not very likely, at least in my opinion. And this is mostly down to a kind of a data set size, training set size issue, right? So there's a lot of kind of arguments you can make about this sort of slide, but generally the idea is that we have these very successful machine learning models that are done by completely different fields, and they have these just insane data sets to work with, right? And then if we go all the way to the right on this slide, we look at what we're actually dealing with, and we consider the PDB huge. It's nothing compared to what DeepMind is working with in OpenAI. And then when we look at the kind of typical drug discovery campaign, it's just infinitesimal, or tiny, I guess. So this is preaching to the choir, obviously, but one idea here is to make science more collaborative. So something like open science drug discovery could be super useful here, right? If we, across the globe, collaborate on a scientific endeavor, in principle, we can reach more together while being more efficient and duplicating less effort. So I'm going to introduce ASAP. Before that, I introduce the kind of thing that came before ASAP, and then I'll talk about the actual blind challenge that we ran together with Polaris. So this is just the general introduction slide. There's a very crowded overview of what the COVID moonshot was. You might have heard of this at the time. You might have participated in it because it was very kind of community sourced. But basically, early 2020, we all know what happened, but at that time, a group of scientists came together from various labs, and they basically made a drug discovery pipeline. This was fragment-based drug discovery, and the idea was, or the dream was, to make this completely open science in multiple fronts. But in general, the idea was we're going to just soak a whole bunch of fragments in SARS-CoV-2 main protease, engage the community to see whether they can come up with good ideas based on those fragments, and we're going to just whip through all of those predictions, make the compounds, elaborate on them further, and all the while share all the data that we generate, which is crazy. Crazy in the sense that it hasn't happened that much before, but also in the sense that the amount of work this actually produces is very, very large. Actually outputting your data in a kind of good way is very, very difficult. So that actually progressed all the way to a preclinical candidate for MPRO, and we've published this in Science fairly recently, so you can read up on that there. Shionogi hasn't helped making that particular preclinical candidate, but I just put the logo there because Shionogi actually now has a marketed drug for MPRO, marketed just in Japan, as far as I know. It's called Encitrovir, and when they published their paper, they actually described how they used the COVID moonshot data while it was being turned out to generate their early hits and their early lead designs as well. So that's just a prime example of how this approach can work. So based on the success of the COVID moonshot, the NIH, so in the US, signed a grant approval for the same group of people to actually do this again, but in a much more formal way. So not just for SARS-CoV-2, but really in the interest of pandemic preparedness. So the idea was to do kind of a similar approach, also do it open science, but do it for other viruses than coronaviruses, but also coronavirus. And also continuously output all of that data. So this is, again, a very crowded slide, but this is kind of the fragment-based drug discovery pipeline that ASAP follows. So you can kind of look at this as the classical pharmaceutical pipeline that is preclinical. So we have kind of hit discovery and target discovery all the way to hit the lead and then lead optimization. And where we wanted to do this particular challenge is in the lead optimization phase of our pipeline. So, as I said, it's kind of insane to be putting out this much data. We could just say, we take all of our internal databases, we zip them up in a zip file and just drop it somewhere on Google Drive, right? But that's just not useful at all. We want to have our science be reproducible and useful to people. So we've actually engaged with a whole bunch of different kind of platforms. One of them is Campbell. I think probably all of you will know Campbell, where we have been uploading a lot of data. We have a lot of biochemical potencies that we're kind of pushing towards Campbell, but that process is kind of slow. So the next 3 or 4 Campbell releases, you can expect thousands of points coming from just us. The PDB, as we all know, is very nice, but it's also kind of difficult to deposit structures into. It takes a while. But we've been churning out a lot of structures there as well. So just last week or 2 weeks ago, we dropped another 600 crystal structures just into the PDB. And then finally, kind of in the spirit of reproducibility, we're working with protocols.io, which is a West Coast-based company that basically allows scientists to write standardized experimental protocols. So these are well, standardized protocols where bench chemists can literally just write down the steps that they're taking for their protocol so that people can easily reproduce these. If you go to our website, ASAP Discovery, you'll be kind of presented with this very large pipeline figure. Usually if you go to a biotech or a pharmaceutical company's website, you'll see this large static image, very snazzy, and it looks kind of like a pipeline. They'll show some targets maybe if you're lucky, and then all the way to preclinical or phase 2 or whatever. And then it doesn't really tell you anything. It just kind of is kind of a bragging thing. But for us, what we've done painstakingly is we've built an interactive pipeline where if you go to our website and you look at all of these individual phases, there are many sub-phases, right? Each of these little blocks you could click, and then you will be directed to where the actual data lives. And the data is curated, and you can freely download. And I think it's under MIT license in general, so you can do with it whatever you want. This all actually leads to something as well. So just a few weeks back, Ed Griffin, who isn't here yet, but he's about to arrive, I think, declared our preclinical candidate for MPRO, which is a dual inhibitor for SARS-CoV-2 and MERS-CoV at the same time. Derek even blogged about it. Very honored. It's a molecule that some of the people that have participated in the challenge that we ran will actually recognize. And we've developed this in such a way that it's resistant to mutations and everything. Very relevant, obviously, if you want to make an antiviral drug. So at this point, you're probably wondering, how are you going to make a marketed drug if all of your data is open, and what do you do with the patents, et cetera, et cetera? That is exactly what Ed is going to be talking about tomorrow. So any difficult questions, you can hold off until when he's given his talk. But I just wanted to bring us back to my first point, which is that there isn't enough data really to make a good machine learning model that's kind of generalizable and can be kind of a turnkey solution to drug discovery. Instead, what basically everyone is doing is training local models. And by that, I mean some company will be generating in-house data, and they just train a model on that in-house data. And you call this a local model because it's trained on the local chemical space. And that works really well, but that model isn't going to be useful for another target, right? Because it's very kind of tailor-made to your own data set. So what we wanted to do with this challenge was we wanted to see what is really the best way to make these local models. How can you get the best bang for your buck and really squeeze out the most of the data that you've been presented with in such a setting. So you remember the pipeline across all of these targets that we do drug discovery on. At the top, you might have spotted the MPRO ones. So we have the program for the dual inhibitor of MERS-CoV and SARS-CoV-2, MPRO, and we decided to basically run a challenge just based on that project. So we've actually teamed up with Polaris, who very conveniently CASP just kind of described everything about. So I'm not actually going to talk that much about what Polaris is. And we actually have just finished that competition as well. So I am going to talk a little bit about the data that we have, but again, I did most of the analysis over the last few days and last week. So this is very hot off the press. We wanted to look at a few different kind of sub-challenges. So biochemical potencies, we all know. It's what most people talk about. It's probably the least important when you're in lead optimization, because you start to think about the harder questions like ADMET properties and what is it going to do when I start giving it to people. That is obviously the middle panel here. We have actually run the challenge on a bunch of those endpoints as well. I have to admit that the analysis of this is still ongoing, so I won't be able to present on that today. And then finally, my personal favorite is ligand posing. So can we predict the ligand pose of some of these crystal structures? And this is very relevant for this particular project because one of the two proteins that we were targeting was the MERS-CoV MPRO protein. And it took really about a year and a half into this project for the crystallography team to actually enable crystallography for that particular target. So we were kind of shooting blind for a year and a half. So we really wanted to see whether something like a machine learning model would be able to have helped us at the time. So you get a lot of kind of questions, right, that you have to think about when you're preparing a challenge like this, because this is kind of a retrospective challenge. How do we split the data? We did it in a kind of 80-20 split, even though it's just training and test. We wanted to have a test set size that is of sufficient size to be kind of ready for rigorous statistics. We did a temporal split, but there were some complications, of course, because drug discovery is very messy. There's stereochemistry, there's remakes, there's overlap between all of these data sets, so it's more or less 80-20, basically. So if we just focus on the timeline first, we really started in January. We'd announced everything the last winter, and then we had about 2 months' time for people to actually put their predictions in through the Polaris platform. During that time, we did these kind of virtual walk-in sessions where people could come into a Zoom room and literally just ask us questions. This was very fun because we had all the crystallographers and the AdMed people and everything, and they could just answer those questions, and we had a lot of nice discussions. And it ended with the disclosure of our compound at ACS this last, 3 weeks ago. And now we're obviously, processing all of those results and working towards a special issue in JCIM together with all of the participants. We saw a really nice distribution of people that actually participated in this challenge, mostly academic. Quite a lot of drug discovery companies actually participated. We saw some software companies, and then we had some people that literally just did it for fun, which was really cool. Then for statistics, I think Cos already mentioned it very briefly. We are doing a kind of Pat Walters-friendly statistical analysis on separating the kind of ranks of all the participants. Pat talks a lot about how he would like models to be compared. He obviously, it's not a very good idea to just slap down a table and have people figure it out for themselves. It's much better to actually do some kind of statistical significance test on whether populations of data can be separated from each other, right? So this is just a very simple kind of high school level ANOVA, where if you look at the right-hand side plot, in principle, the blue point with its confidence bounds is the top performer in this particular case. All the gray ones are not statistically distinguishable from the top performer, so they are the same. And then the red ones are distinguishable, so they are worse than the top performer. We have one problem here, which is we don't really have good confidence estimates from our participants, right? I think if we asked people to put in their own confidence metrics, they would probably all just give super high confidences, and that would be useless. So we ended up having to bootstrap everything, and bootstrapping is bootstrapping is notorious for being a kind of underestimation of the confidence, but still, it's everything that we were able to work with. So one additional thing that we added on top of Pat's idea is that we have the CLD, the compatibility display, which is essentially just a way to also vertically indicate the kind of order of the ranking. So in this particular example, if we have entry rank 1, 2, and 3, 1 and 2 would be equivalent, and the third one would be number 2, right, in the ranking. So this is just to show you what the biochemical potencies would look like. This is an extremely complicated plot. We have separated it by SARS-CoV-2 predictions and MERS-CoV predictions. So we have Kendall's tau on the x-axis, which is just the ability to rank things. If we have 1.0, it means that the test set was perfectly ranked. But you can see that for, especially for SARS-CoV-2, we hit about a 65% accuracy in ranking. It's actually a little bit more interesting to overlay these. So there's a little bit of obfuscation. So don't pay attention to the y-axis anymore because the methods are now just kind of mixing together. But you can very clearly see that there's a clear shift, right? You can see that the orange ones, which are the Merskov predictions, are just for, I think for every entry are markedly worse than for SARS-CoV-2, which makes sense because there's so much more data available for SARS-CoV-2. We also saw very clearly that for these local models, for potencies, the machine learning and neural network methods are working much better than the kind of old school QSAR and physics-based methods, right? So I think someone even did 300 calculations, but that's actually my background, and I can tell you the chemistry is way too diverse to do anything like that. So this actually is kind of a nice confirmation of what everyone is talking about in the field of machine learning and CompChem. So because drug discovery is very messy, the actual analysis of all of this is also going to be very messy. So here's a first attempt where I've split the potency performances into kind of time bins, right? So we see the very old compounds in general for MERS-CoV-2, so blue, the absolute error of the prediction per entry is very high, and then for the younger compounds, so really in 2024 mostly, we see that it gets much better. So for SARS-CoV-2 and Pro, it's more or less stable the whole time, but the early on MERS-CoV compounds were predicted much poorer by participants, which is a really interesting finding, right? And I don't really know why yet, but my suspicion is that it's because there was a kind of consolidation of the chemical space that we were exploring towards the end of these two projects, where towards the end we were much more focusing on ADMET, so there are much more measurements of compounds that were really in a very tight chemical space. So as I said, my favorite is post-prediction. So I wanted to talk about that a little bit as well. For this particular sub-challenge, we had some baselines that we just did ourselves because we wanted to see how well they would kind of hold up to what our participants would come up with. So we did two things. At the top is our own thing that we did, literally. Well, we run this toxicology campaign where we did a kind of informed docking approach that basically took from a reference set or a training set, we took the 5 closest compounds and just in 2D, so based on the maximum common substructure. And then we kind of redocked those, the new query compounds using that reference. And then we just took of the 5, the compound that had the highest or the best docking score. Then secondly, we have the blind docking baseline, which is literally just, here's a binding pocket, jam it in. That's kind of what everyone does most of the time. So when someone talks about Janina or something, pronounced Nina, then that's usually what they're doing. So this is what that looks like. So you see a lot more kind of spread than with potencies. And here it becomes really cool, right? So we have the blind docking, which is really bad. On the x-axis, by the way, we have our success rate, which is just for a particular participant, all of their predicted poses. Every pose that was within 2 angstroms RMSD of the true pose is a successful pose. And then we just say, what is the percentage per participant of successful poses versus all of the poses that they submitted. Blind docking, it's basically random, whereas the informed docking is actually really good, right? So we're pretty happy that we didn't just generate random numbers for 3 years. But you can see that there's a few models actually here that are even better than what we've been doing. So we're going to do the same overlay. And you can see, again, very clearly that MERS-CoV, so the orange colored dots, are performing worse generally than SARS-CoV-2. SARS-CoV-2, I should say, there was much more in the PDB at the time than just the training set that we gave participants. But it's very cool to see that these top models are almost invariant to which target. And it's even more special because all of these top models are co-folding methods, right? So these are all AlphaFold 3-esque models. Most of these entries actually went into the training set, cleaned everything out, and kind of decided what they wanted to pay attention to, fine-tuned their models on that sub-selection, using a validation set within the training set as well. And then sometimes they had some like clever post-processing where they kind of minimized or went back into the training set and kind of adjusted some poses here and there. Yeah. So this is really, for me, the coolest finding right now, where this would have been insanely useful at the time during our campaigns. So just some first conclusions here. We have tried as closely as possible to mimic a kind of drug discovery campaign for this challenge. And we saw that it really interested a lot of people, right? We've had about 100 participants actually participate in this challenge. Everyone was super engaged and enthusiastic, and everyone came from different angles with different expertises that they wanted to apply, which is really cool. Making it very close to drug discovery also increases the complexity of actually analyzing this data. We will be writing a paper that hopefully describes as well as possible all of the findings that we've observed here, but that is definitely something that we hadn't really realized beforehand, that it would just become very difficult to kind of untangle all of the stuff that would come out of this challenge. We saw that for the potencies, the machine learning and neural network models outperform everything else, and then that these co-folding methods are actually really, really good when handled properly. We saw for both these sub-challenges that I showed you, we saw that MERS-CoV-MPRO was generally more difficult to predict on than SARS-CoV-2-MPRO, which is nice because that was the case for us as well, so we weren't just being stupid for 2 years. And then finally, I didn't really talk about it, but the Polaris platform, I'm actually convinced was really the main driver behind the engagement of the community because of its ease of use, right? So CASP showed a little bit of code. This is a little bit more code, but it's actually amazing that you can go in 4 lines of Python from downloading the competition, splitting the test set and the training set, making your own predictions. That's obviously a lot more lines of code, but nothing to do with Polaris. And then the 4th line is literally just submitting everything. So with my ACS brain active, I made this slide. You're obviously all going to be in this room probably, so well, pay extra attention. Cas already talked. Carmen is going to talk later today about everything OMSF. So if you like open software and if any of these things ring a bell, then definitely check those out. Ed is going to be talking about our patent strategies tomorrow. And then Danny participated in this challenge as well, and he is speaking on his entry to last year's cash challenge, I think. So all the ASAP people, have to thank them. Almost done. So and then the Polaris people for actually enabling us to run this challenge. Hugo up at OpenAtMed for just being a partner in crime with me running this challenge, and then finally all of the ASAP people that actually ran and orchestrated all of these experiments. And thank you for your attention.
Thank you so much, Jenke.